Hi, this is Mohammad Kamruzzaman. I'm a graduate student from Department of Biology and I'm doing my research on study of soybean symbiont Bradyrhizium japonicum ECS sigma factors in response to desiccation stress. Just to give you the idea of the background, we know nitrogen is a major component required for plant growth, and this is why farmers use chemical nitrogen fertilizer. But the problem is it contaminates the environment and the water system. So to solve this problem, symbiotic nitrogen fixation is a good option because it you it uses the soil rhizobia, and soil rhizobia they can form symbiotic relationship with soybean and fix atmospheric nitrogen in the form of ammonia so that plant can uptake it. We also know that drought is a limiting factor for the soybean production in the United States, and not only the soybean but also its symbiotic partner rhizobia affected by the drought or desiccation stress. Considering this situation, we started working on the ECF sigma factor, where ECF sigma factor itself is a transcription factor that can activate the transcription of stress responsive gene during the stress. So in the figure 1, it's actually showing an example of the drought stress soybean field, and figure 2, it's a model of ECF sigma factor involves signal transduction in the presence of stress. That means if the stress is present, the anti-sigma factor release the ECF sigma factor, and ECF sigma factor activate RNA polymerase, and RNA polymerase activate the stress responsive gene. So in this case, the stress responsive gene is basically the trehalose biosynthesis gene. So the trehalose is a disaccharide osmoprotectant that help the bacteria to survive during the desiccation stress. So during the for the materials and method part, we selected the gene based on the protein sequence similarity with a known ECS sigma factor that function already identified previously. So here we considered the BLR7797 or ECFG because it has function on desiccation and symbiosis. So after selecting the gene, so we selected two gene, one is the BLL3014 and second one is the BLL2628. So after selecting the gene, we make the knockout mutant by site specific mutagenesis and replace it with the antibiotic resistant gene, which is kanamycin. So after making the mutant, we did the desiccation assay. We grow the bacteria in the media, suitable media, trap them in the filter paper, and then keep them in the desiccator with two conditions. One is the hydrating, another one is the desiccating condition. And then after certain time point, we again resuspend the cell and did the CFU count. So this is actually the result of the mutant conformation. Like for figure 6, it's the conformation gel image of BLL2628 and the 3014. So figure 7 actually showing the percent survivability of between the wild type B japonicum USDA 110 and the mutant. So the A is comparison of USDA versus the first one 2628 mutant, and B is USDA versus 3014. So in both cases, x-axis showing the time point, left y-axis showing the log CFE value, right y-axis showing the percent survivability. So the black dot here is the showing the percent survivability, and the blank dot showing the log CFE value. Green line is for the mutant, red line is for the wild type. So in both cases, we can see that the mutant is surviving less than the wild type. So in conclusion, we can say that a mutant surviving less than the wild type up to 96 hours, which is statistically significant. And this result indicate that 2 sigma factor might have essential role in the transcription of trehalose biosynthesis gene. For the future direction, we can study physiological characteristic of the mutant, especially the symbiosis and the nitrogenous activity. And also we can study interaction of the ECF sigma factor with regulatory gene and their interaction during the desiccation stress. Thank you all for your patience and giving me the opportunity to present my work.